Looking deeper at blood types, we'll go back to my family for an example. My mother has type B blood. My brother has AB type blood, and I have A type blood. My father said that he wasn't 100% positive about his blood type, but that he thought he remembered it being A type blood. And I said, well, I'll take a look at that. And yeah, it seems like that's probably pretty plausible. Both my brother and I have an A-type allele, which is coded for by a dominant allele. If my mother had that allele, then she would have an A represented in her blood type. So we had to get it from somewhere. If we didn't get it from her, we must have gotten it from him. So I think his memory that he had A-type blood is probably accurate. And I'll just take it for granted that that's what he has and not AB-type blood for the rest of this question. Now, if we wanted to figure out what the genotype of everyone in my family is for their ABO blood group, my brother's is the easiest to work out because both A and B-type blood are coded for by dominant alleles. If he has both of them, he must have one of each. So his genotype is capital I-A, capital I-B. He's actually heterozygous, but he has two dominant alleles. It's in codominance that can happen. Everyone else, we know half of their genotype right off the bat. We know that they either have the A dominant allele or the B dominant allele, so I'll pop those in here. Now, you might say, well, that's all the information we can really figure out here. That other allele, it's mysterious. However, we can figure out a little bit more about this problem, because if my mother's second allele was dominant IB, then she would have had no choice but to give me one of her two dominant IB alleles, and then I would have a B represented in my blood. She can't possibly have a dominant IA, or else she would have an A represented somewhere in her blood. So that leaves us with only one possibility. She must have a recessive I as her second allele, and I must also have inherited that recessive I for my second allele. Now, my father, we can't be 100% certain for him, because he did give both of his sons this dominant IA allele, but we could have both gotten copies of this same one. We don't know that his second allele matches, but he just as easily could have a recessive allele at that position. We just don't know. If they were to have more kids or something like that, if we had some other sort of sample size, we could tell. But for right now, we have to leave it that he's either one or the other. When you're unsure in problems with this, the common way to write it is that you know this, but that the second letter is a mystery, so you just leave it as a blank. In polygenic inheritance, more than one kind of gene can affect the phenotype of the offspring that are created. So here we have an example where there are 3 different genes, A, B, and C, that affect skin pigmentation at birth, how much melanin is produced. Each of them can either turn up melanin production or turn down melanin production. If you have all 3 genes turned up, then you make a lot of melanin. You end up with a very dark skin pigmentation at birth. If you have them all turned down, you'll end up with a very light skin pigmentation at birth. Here, in the P generation, we have 2 individuals that are homozygous for all of these genes. So this person is homozygous recessive for all these genes. This person is homozygous dominant for all of these genes. If they were to have offspring, all of their offspring would be heterozygous for each of these traits. A monohybrid cross is when we cross 2 heterozygotes for a single gene that we're considering. We also talked about dihybrid crosses, where we cross two different people that are heterozygous for two different genes. This would be a trihybrid cross that we're doing. It's just that all of the genes that we're concerned about here happen to affect the same phenotype. In this situation, you would get a very small proportion of the offspring with nothing but recessive copies of the gene. A very small proportion also you would get with nothing but dominant versions of the gene, and you would see every single skin pigment in between along the way. This is a hallmark of polygenic traits, is that you don't just have a binary situation, very tall or very short. You have every possible state in between the two extremes. Another situation is called pleiotropy. This is kind of the opposite of polygenic traits. Polygenic traits is you have many genes that affect one phenotype, whereas in pleiotropy, we have a single gene which is going to affect many different traits. So, one example of this would be in chickens. Take a look at this very fabulous looking specimen of a chicken right here. It has the so-called frizzle gene. That gene causes their feathers to grow in a little bit curly, so they come in looking like they just got out of the hair salon with lots and lots of mousse in their feathers here. However, that same gene also affects a few other traits in that organism. So they're going to have abnormal body temperatures. They'll have higher metabolisms, a higher rate of blood flow, and they generally lay fewer eggs. So that is pleiotropy, one gene affecting multiple different phenotypes. We also talked about linked genes a little bit. So if you have a chromosome, and it's got gene number 1 on one part of it and gene number 2 on another part of it, then that means gene number 1, number 2, that means you'll probably inherit both of those genes if you get one of them. Not always, though, because crossing over sometimes separates two alleles from each other. But it turns out that some genes are physically linked together, so you will disproportionately inherit them at the same time. One thing that some Russian scientists tried a while back was to take the European red fox, and they wanted to breed them for a calmer temperament. They wanted to make them a little bit more friendly. The way they do this was by measuring what's called flight distance. It's how close you can get to an animal with a bit of food before it runs away. If it'll eat it out of your hand, its flight distance is 0. But if they let you get a foot away, that's one thing. A yard away, they're even more skittish. If they run away as soon as they see you, that's really skittish. So they would breed together foxes, and then they would select the pups that had the lowest flight distance, the ones that were the most comfortable coming up to the humans and interacting with the breeders. That was the only trait that they were really interested in, because they just wanted friendlier foxes. Maybe they were trying to sell them, I'm not really sure. But as they did that, they selected for this behavioral trait. Other traits started to come along because it turned out that they were linked to the kinds of genes that they were looking for. So the ears became a little bit rounder and a little bit floppier. The coat color started changing colors. The tail started curling up at the back, and they started to behave and look a lot more like modern day dogs do. They became super friendly, dog-like foxes here. So it turned out that the genes for behavior were actually linked to some of the genes for coat color and ear shape and all of these other traits as well. Now, because of crossing over, which you'll remember from meiosis, It's not a guarantee that any two genes that are connected on the same chromosome will remain linked when they're inherited in the next generation, because mom and dad's chromosome number 1, they will wrap their chromatids around each other during prophase 1, and they'll swap genes sometime. The closer two genes are together on the chromosome, the more likely it is that they will be inherited together, because the cut on these chromosomes where it's exchanged is random. So if it happens in between these two genes, they will separate. If it happens on the far side of one of these genes, or right here on the tip, they will be inherited together. Now, another example of where something like this occurs is called epistasis. In epistasis, a gene at one location alters how a gene at another location will express its phenotype, how it will be represented in the organism. They're not all contributing in the same way as I was just talking about with skin pigmentation, but they might actually alter each other. In Labrador retrievers, dogs, coat color depends on two different genes. One gene determines the color of the pigment, K, dominant K for black, and recessive K for brown. Now, even that is a simplification. So, for example, my dog Edward here has a brindle coat, which actually turns out to be represented by another kind of recessive gene for coat color at this same locus. So he has two recessive genes for brindle. The other gene that is affected here, we given the letter designation E, and that determines whether or not that color will actually be produced and show up. So what kind of color might be represented is at the K locus, but whether or not we actually get the color is at the E locus here. So here is a dihybrid cross of two Labrador retrievers, both of whom have a dominant K for black fur, a recessive K for brown fur, a dominant E for pigment production, and a recessive E for no pigment. Now, because both of these dogs have one dominant allele for both of these pairs, they end up with the dominant phenotypes. That is, black pigment, and the pigment is indeed being produced. So we have two black-furred dogs here. If these dogs were to reproduce together, this is the Punnett square that represents the different kinds of offspring that they might have together. So what would the phenotypic ratio of these dogs end up being? Well, here we have 1, 2, 3, 4, 5, 6, 7, 8, 9 dogs that have both dominant traits represented in their phenotype. There's pigment being produced, and the pigment is black. We have 1, 2, 3 dogs who have the recessive gene for coat color, that is, they have brown coats, but they do have the dominant phenotype here for pigment production, so that you actually see the brown coat. Now, here's where it varies a little bit from what we've seen before. These 3 dogs, 1, 2, and 3, all have the dominant genotype, which would indicate that they have black fur. However, they have the recessive genotype, the homozygous recessive genotype, which prevents that pigment from being produced. So they end up as golden retrievers, as blonde dogs here. Normally, the dihybrid cross ratio is 9 to 3 to 3 to 1. This final dog here is the only one that's homozygous recessive for both of these genes that we're looking at. But because this is a case of epistasis, where one gene affects another, the fact that it is homozygous recessive for this trait is really the only thing that's important in determining its phenotype. As a result, these 4 dogs will look indistinguishable from each other, at least as far as their coat color is concerned. So instead of 9-3 to 3-1, those last 2 get combined, and we end up with a 9-3-4 ratio for this particular dihybrid cross. Epistasis changes the rules a little bit, just like incomplete dominance and codominance do in monohybrid crosses. Another exception to Mendel's rule. It shouldn't matter for most traits, for most genes, whether you got a particular gene from your mother or your father, or whether you're male or female, or what happens. But some genes are sex-linked. Human beings have 46 chromosomes total. These chromosomes occur in pairs. 22 of these pairs are what we call autosomes, your non-sex chromosomes, and everyone has 2 of each of these, unless you have some sort of chromosomal condition. But the last pair, what we call the sex chromosomes, are either your X or Y chromosomes. Here's what they look like. This is the X chromosome right here, and this is the Y chromosome here. The Y chromosome is a good deal smaller than the X chromosome, which makes sense because human biological males have 1 X and 1 Y chromosome, whereas females have 2 X chromosomes. So only 50%, a little bit less than 50% actually, if you look at the numbers, of the human population has a Y chromosome. Therefore, you don't want to put any genes that are particularly important to the survival or development of an organism on that chromosome. If there's a gene that's really important for the development of lung tissue, for instance, you don't want to put that on a chromosome that only half of your population is going to get. Everyone needs that gene. So the Y chromosome only has a few genes on it which are related to human biological maleness. Now, just like with any other set of chromosomes, sometimes you have genes which code for certain types of conditions that pop up on the X or Y chromosomes. We call these sex-linked genes. Colorblindness, red-green colorblindness, is an example of this. This occurs on the X chromosome. So if we were to scan through here, we would find somewhere on this X chromosome there would be a gene for colorblindness. Now, when you have sex-linked recessive genes that code for some condition, that condition will usually disproportionately affect biological males as opposed to females. Now, that seems odd, that no matter which sex chromosome it's on, it's going to disproportionately affect males, but the math makes it all make sense. Let's say that I have some condition which is recessive, and it occurs in 10% of the population. Usually, human genetic conditions are recessive, and usually they're quite rare, so 10% would actually be pretty big. So 10% of all X chromosomes out there in the population have this condition on them. Every biological male gets 1 X chromosome, so they have a 10% chance, 1 in 10, of inheriting this condition. However, since it's a recessive trait, for females to inherit that same condition, they would need 2 of those affected X chromosomes. So 10% odds that their first X chromosome is going to have that condition, but 10% odds that the second one will. You multiply that together and you find that only 1% of the female population would end up with that condition, which is 1/10 as many as the male population. So that's what happens if a sex-linked gene, a recessive sex-linked gene, is on the X chromosome. But what if it's on the other one? What if it's on the Y one? Well, if it's on the Y chromosome, it can only affect human biological males because the females obviously wouldn't have one in order to get the condition. So no matter which sex chromosome it's on, generally sex-linked genes tend to affect men if it's some sort of condition that we're concerned about. Colorblindness is an example. So if you're red-green colorblind, you can see the value, the light and shade on an object just fine, but you cannot determine the hue of the light coming off it. Green and red look pretty much the same, so this is how a colorblind person might see the world. Now, this occurs on the X chromosome, and generally it affects men more often than it affects women. Continuing from our discussion of colorblindness from the previous slide, down here we have a classic test for colorblindness. There is a number that is hidden inside this pattern of dots. If you cannot tell distinguish the difference between red and green, it would be very difficult to be able to read this number because the pattern of dark and light doesn't really matter here. Only the hues of these dots actually matter. If you can tell the difference, then you should be able to see that there is a 7 written inside the circle. And for those of you who cannot, I don't know if this is frustrating or helpful, but it's right there. Up here, we have a story about a family over a few different generations, and kind of an example of why colorblindness disproportionately affects males in the family as opposed to the females. So up here, we have a colorblind male. He's got the recessive version of this gene on his X chromosome, marries a woman who has no genes for colorblindness. She has only the color-seeing copies of these genes, the dominant version. Well, since this father is going to give his X chromosome to his daughters, and each daughter is going to inherit a functional copy of the gene from their mother, they are going to be born able to see color. They can see it just fine. However, they are carriers for the condition. The sons, who only inherit their X chromosomes from mom, they'll end up not colorblind at all. Now let's say that one of these daughters grows up and she marries a male who has no genes for colorblindness in his chromosomes here. So it's the same thing as before. We have one individual who has a gene for colorblindness, one individual who does not, and they're having offspring together. But now we can see that because 50% of the daughters are going to get her first X chromosome with the functional copy, 50% of her daughters will not receive any colorblindness gene at all. And since 50% are going to get this copy of her X chromosome, 50% will become carriers. But it's in the sons that it becomes a little bit more interesting. Every son is going to get the Y chromosome from the father. However, they will get their X chromosome from their mother, either the functional copy or the non-functional copy. So 50% of this woman's sons will be born colorblind, as a matter of fact. Then, if we take this colorblind male and he grows up and he wants to have kids of his own, and he marries a woman who happens to be a carrier for colorblindness, she can see just fine, but she's a carrier for the condition, then, again, 50% of their sons are going to be colorblind, but only in this situation can we actually start to see colorblind females start to pop up. So there are colorblind females, they're just less common than the colorblind males.